Hello everybody, I'm Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we're going through the ASCP reading list together. We've already finished the Hematology and Chemistry modules, and now we're moving into the Microbiology module. This first video is an essential introduction you must understand before we deep dive into the details. Think of it as your foundation. Without this, everything else won't make sense. Let me start with something that might blow your mind. You are not alone. Right now, as you're watching this video, there are billions, actually trillions, of tiny organisms living in and on your body. You're essentially a walking ecosystem, a mobile planet hosting an incredible diversity of microbial life. Our mission in this introduction is to understand this hidden world. We'll map out who's living there, the good guys we call normal flora, and the troublemakers we call pathogens. We'll explore the main players, bacteria, viruses, fungi, and parasites. We'll see how our friendly microbes actually protect us, and then how pathogens launch their attacks against us. This is essentially a survival guide for understanding planet human, and crucially, what all this means when you're trying to diagnose and treat a patient in the clinical lab. Let's start with a basic census. Who are these microscopic residents? How do we even classify them? The most fundamental biological division is between prokaryotes and eukaryotes. And before we go any further, we need to talk about taxonomy, the systematic science of naming and classifying organisms. This is absolutely critical because it ensures that a microbiologist in Tokyo and one in London are talking about the exact same organism when they use a specific name. The bedrock of taxonomy is the binomial system, 2 names for every organism. First comes the genus name with the first letter always capitalized. Second comes the species name, always lowercase. Here's a key detail you need to remember for reports and documentation. The complete 2-part name must be in italics when typing, or underlined when writing by hand. For example, the bacterium that causes pneumonia is written as Streptococcus pneumoniae, or you might see it shortened to S. pneumoniae after the first mention. Getting this naming convention right is step 1 in avoiding clinical mistakes. The taxonomic hierarchy goes: domain, kingdom, phylum, class, order, family, genus, and species. For bacteria specifically, family names always end in ace, like Enterobacteriaceae. That's a pattern you should recognize. In medical microbiology, when we say prokaryote, we're almost always talking about bacteria, specifically eubacteria. The defining features of bacteria are size, small, typically 0.2 to 2 micrometers. Structure, simple, lacking internal organization. No nucleus, DNA floats freely in a region called the nucleoid. No membrane-bound organelles. No mitochondria, no endoplasmic reticulum. Cell wall, they have a rigid cell wall that's crucial for structure and survival, and this wall is often a target for antibiotics. The other major group we deal with are eukaryotes, much more complex and larger, generally 10 to 100 micrometers. This group includes fungi. These include molds and yeasts. They're eukaryotic but can't photosynthesize. They usually live off decaying matter, we call them saprophytic, and reproduce using spores. Fungi can cause everything from annoying infections like athlete's foot to really serious systemic infections, especially in immunocompromised patients. Protozoa, these are single-celled eukaryotes that don't photosynthesize. Many are free-living, but the parasitic ones cause significant disease. They can infect virtually any tissue. Think malaria caused by Plasmodium, or giardiasis caused by Giardia. Helminths, these are the worms, multicellular parasites including tapeworms, flukes, and roundworms. You can actually see many of them without a microscope. They survive by eating or absorbing nutrients from their host. Now we come to the real oddball, viruses. They break all the rules. They're not even truly cells, and whether we call them alive is actually debatable. Here's what defines a virus. Obligate intracellular parasites. They absolutely must get inside a living host cell to do anything. Genetic material, just DNA or RNA, never both, wrapped in a protein coat. No cellular machinery, no ribosomes, no energy production systems, nothing. Complete dependence. They must hijack the host cell's entire factory, its ribosomes, enzymes, and energy, to replicate. Think of them as microscopic pirates with the blueprint for making more pirates, but they need to steal someone else's entire ship and crew to actually build anything. The 3 domains of life are bacteria, archaea, and eukarya. Now, archaea are also prokaryotes, but we rarely encounter them clinically. They're extremophiles, organisms that thrive in extreme environments like boiling hot springs or incredibly salty water. While they're biochemically fascinating, they typically don't cause human infections. The clinical microbiology lab focuses overwhelmingly on bacteria and pathogenic eukaryotes. Modern identification methods. Historically, we relied heavily on phenotypic characteristics, what the organism looked like and what it did. This included microscopic morphology. What do the cells look like under the microscope? Colony characteristics, size, texture, color on agar plates. Metabolic activities. What can they eat? Do they need oxygen? Gram stain, the classic phenotypic test we'll discuss in detail later. The modern view recognizes that relying on observable traits alone isn't enough for accurate identification, especially with closely related species. So now we use polyphasic taxonomy, which combines genotypic data, genetic information, phenotypic observations, visible characteristics, phylogenetic relationships, evolutionary connections, chemotaxonomic methods, chemical composition. For a long time, the gold standard was sequencing the 16S rRNA gene. This gene is highly conserved, so it helped map deep evolutionary branches. But here's the limitation. For distinguishing very similar species or different strains of the same species, 16S just doesn't have enough detail. It's like reading one paragraph of a book when you need the whole story. We've now moved to next-generation sequencing methods that give us the whole book. Average nucleotide identity, ANI. Instead of just one gene, ANI compares thousands of genes across the genome, looking at average similarity across huge chunks of DNA. Genome-to-genome distance, GGD, a computational approach using whole genome sequences to calculate an overall similarity score between organisms. The key point: we can now sequence entire genomes quickly and cheaply, which has revolutionized how accurately we classify microbes. This has huge implications for tracking disease outbreaks and understanding pathogen evolution. A game-changer in modern labs is MALDI-TOF-MS, matrix-assisted laser desorption/ionization time-of-flight mass spectrometry. Yes, it's a mouthful. Essentially, it creates a unique molecular fingerprint of the organism by analyzing its most abundant proteins and peptides. It's fast, provides a chemical profile, and works excellently alongside genomic and phenotypic data for robust identification. Here's something that changed everything we thought we knew. For decades, we relied on growing microbes in petri dishes on culture media. But many bacteria simply won't grow under standard lab conditions, so we got a really skewed picture of microbial diversity. The revolution came with DNA sequencing technology. Suddenly, we could sequence all bacterial DNA present in a sample without needing to culture anything. The results were shocking. Example: we used to think Staphylococcus epidermidis and its relatives made up about 90% of aerobic bacteria on skin, based on culture results. But DNA analysis showed S. epidermidis is actually only about 5% of total skin bacteria. The skin is an incredibly diverse ecosystem with loads of different species we simply weren't seeing before. We were essentially blind to 95% of what was actually there. What is normal flora? Let's focus on the overwhelming majority of microbes, the ones that are supposed to be there. We call them normal flora or commensals. Commensal literally means dining together. They live with us, often benefiting us. Critical point: it's essential to remember where they should be. Your skin, mouth, and gut are colonized with normal flora starting almost immediately after birth. But your internal organs, blood, brain, spleen, lungs, are meant to be sterile environments. Skin flora. Key players on skin. Staphylococcus epidermidis. Propionibacterium acnes, lives deeper in hair follicles where there's less oxygen. Here's a crucial concept about normal flora. S. epidermidis is usually harmless on your skin surface. But if it gets somewhere it shouldn't be, like colonizing a plastic catheter or an artificial joint, it can enter the bloodstream or deep tissues and cause serious infections. It's all about location. A benign resident in one place becomes a dangerous intruder in another. Oral flora. Streptococcus mutans is a major player in forming dental plaque. Again, location matters critically. If you have dental surgery or even vigorous tooth brushing, S. mutants can get pushed into the bloodstream. In someone with a damaged heart valve or an artificial valve, S. mutants tends to stick to those damaged surfaces and can cause infective endocarditis, a potentially fatal condition. A harmless plaque bacterium becomes a potential killer when it relocates. Intestinal flora. This is where the microbial population explodes. The density in the large intestine is astronomical, 10 to the power of 11 bacteria per gram of fecal content. That's 100 billion bacteria per gram. Composition. Over 99% are strict anaerobes. They die in the presence of oxygen. Bacteroid species dominate. Facultative anaerobes like Escherichia coli are also present. E. coli is interesting. Everyone's heard of it, but it's actually less than 0.1% of total gut flora. However, because the overall bacterial numbers are so massive, even 0.1% represents a huge population. And because the gut exit is anatomically close to the urinary tract opening, especially in females, coli easily gets displaced and can travel up the urethra, making it the most common cause of urinary tract infections. Vaginal flora. The key players for maintaining vaginal health are Lactobacillus species. They ferment glycogen, producing lactic acid, which keeps vaginal pH low and acidic. This acidic environment is protective. It inhibits the growth of many potential pathogens. What happens when balance is disrupted? If the lactobacillus population drops, maybe due to antibiotic use, the pH rises and becomes less acidic. This creates an opening for other microbes that are normally present in small numbers to overgrow. A classic example is Candida albicans, a yeast. When the lactobacillus shield is down, Candida multiplies, leading to yeast infections. Why do we tolerate trillions of microbes? They provide major benefits. Competitive exclusion. They physically block pathogens from getting a foothold by occupying space and using resources. Antimicrobial production. Many produce bacteriocins and other compounds that actively kill or inhibit competitors, chemical warfare on our behalf. Immune system development. Early colonization is crucial for stimulating and training the developing immune system, teaching it to distinguish friend from foe. Nutrient provision. They provide vitamins we can't make ourselves, like vitamin K, and help digest complex carbohydrates we couldn't otherwise break down. It's truly a partnership, a symbiotic relationship. The Gram stain, first line of defense. Even with all our modern technology, the Gram stain is often the very first diagnostic step. It's quick, cheap, and provides a massive amount of information immediately. It divides nearly all bacteria into two major groups based on how their cell wall reacts to a series of dyes. The entire test hinges on the peptidoglycan layer. Gram-positive bacteria. Characteristics. Thick peptidoglycan layer, like thick mesh armor. Crystal violet retention. The thick layer traps the crystal violet iodine complex. Alcohol resistance. Even when washed with alcohol decolorizer, they hold the purple dye. Color. Deep purple or blue under the microscope. Additional components, teichoic and lipoteichoic acids in the wall. Gram-negative bacteria. Characteristics. Thin peptidoglycan layer. Outer membrane, the distinctive feature, containing lipopolysaccharide, LPS, decolorization. Alcohol damages the outer membrane, and the thin peptidoglycan can't retain the purple dye. Counterstain. After washing, they pick up safranine and appear pink or red. This is crucial clinically. LPS is also called endotoxin, and it's incredibly potent. When gram-negative bacteria die and break apart, perhaps killed by antibiotics or the immune system, they release massive amounts of LPS. This LPS is the main driver behind severe symptoms of gram-negative sepsis and septic shock, widespread inflammation, dangerous drops in blood pressure, and potential organ failure. Just seeing pink rods versus purple cocci on a Gram stain immediately changes your clinical thinking about potential for rapid deterioration and guides urgent treatment decisions. Not all bacteria follow Gram stain rules. The most important group of outliers are acid-fast bacteria, which include Mycobacterium tuberculosis, TB, and Nocardia species. Why they're different. Unique cell walls, very high lipid and wax content, especially mycolic acid. Gram stain failure. The waxy coat blocks standard Gram stain dyes, appearing as faint, ghost cells, or nothing at all, a dangerous false negative. Special staining required. Acid-fast stain uses strong dye, carbol-fuchsin, often with heat, to penetrate the waxy layer. Acid-alcohol resistant. After staining, they resist decolorization with harsh acid alcohol, staying pink/red against a blue background. Clinical implications. That waxy wall makes these organisms incredibly tough. They grow slowly, resist drying, and are resistant to many disinfectants and common antibiotics. This explains why treating TB requires multiple drugs for many months. For bacteria to grow, they need 3 non-negotiable things. Carbon source. Carbon is the backbone of all organic molecules, makes up about half their dry weight. Nitrogen source. Absolutely essential for making proteins and nucleic acids. Energy source, usually ATP to power all cellular processes. Almost all human pathogens are heterotrophs. Unlike plants or some environmental microbes, autotrophs, they can't use CO2 from air as their carbon source. Heterotrophs must consume organic compounds like glucose or amino acids for both carbon and energy. They essentially eat like we do. How bacteria handle oxygen is hugely important. especially for lab culture. Obligate aerobes absolutely require oxygen use aerobic respiration for energy. Example, Pseudomonas aeruginosa. Obligate anaerobes. Oxygen is toxic to them, only grow in complete absence of oxygen. Require special anaerobic jars or chambers. Examples, Clostridium difficile, Bacteroides fragilis. Facultative anaerobes, largest clinical group, prefer oxygen because aerobic respiration yields more ATP. Can switch to fermentation if oxygen isn't available. Flexible survivors. Examples, E. coli, Staphylococcus aureus. Microaerophiles, need oxygen, but only at low concentrations, 5 to 10%, normal atmospheric oxygen, 21%, is harmful. Example, Campylobacter jejuni. Critical point, if you try to grow Campylobacter in regular air, you'll fail. Capnophiles, require increased CO2 levels, 5 to 10%, for optimal growth. Example, Neisseria gonorrhoeae. The takeaway, getting atmospheric conditions right in the lab is absolutely critical. You'll miss the pathogen if you don't provide the right environment. Energy production, fermentation versus respiration. Fermentation, anaerobic process, no oxygen required, partially breaks down sugars to get small amounts of ATP, produces characteristic waste products, acids, alcohols, gases. These waste products are used for laboratory identification. Oxidation, aerobic respiration, requires molecular oxygen as final electron acceptor, breaks glucose down completely to CO2 and water, generates large amounts of ATP, much more efficient, high-energy pathway. Understanding whether a bacterium ferments, oxidizes, or does both immediately narrows down identification possibilities. The basic prokaryotic genome is relatively simple compared to ours. Usually one large circular chromosome, located in the nucleoid, not enclosed in a nucleus. Haploid, one copy, unpaired. Double-stranded DNA, extensively folded and supercoiled. This structural simplicity allows bacteria to replicate incredibly fast and respond quickly to environmental changes. Beyond the main chromosome, bacteria have extrachromosomal mobile genetic elements, the mobilome. This is like their removable storage. Plasmids, small, circular, double-stranded DNA molecules, replicate independently of the main chromosome, often carry specialized genes, antibiotic resistance, toxin production, like trading cards for survival traits. Transposons, jumping genes, DNA segments that can cut or copy themselves and insert into different locations, can jump between plasmid and chromosome, or plasmid to plasmid, carry genes for their own movement plus additional genes, resistance, virulence, make useful or dangerous genes incredibly mobile. The combination of plasmids and transposons means resistance and virulence genes are portable. They can move within a cell and between different bacterial cells. Bacterial gene expression is incredibly fast because transcription, making RNA from DNA, and translation, making protein from RNA, happen simultaneously in the cytoplasm. As messenger RNA is being made off the DNA template, ribosomes can immediately jump on and start building proteins. This means bacteria can sense an environmental change, like a new antibiotic, and start producing defensive proteins within minutes. A specific detail, bacteria use a modified amino acid, N-formomethionine, as their protein synthesis start signal. This is different from our cells, which use regular methionine. This difference is a perfect drug target. Some antibiotics work specifically by blocking this bacterial starting mechanism, stopping protein production completely. Bacteria share genetic information between cells through three main mechanisms. 1. Transformation. DNA scavenging. Bacteria take up naked DNA fragments from the environment, released from dead bacterial cells that have lysed. Some species, Haemophilus, Streptococcus, are naturally competent at this. 2. Transduction. Transfer mediated by bacteriophages, viruses that infect bacteria. Phage accidentally packages bacterial DNA instead of viral DNA. When it infects a new cell, it injects the previous bacterium's DNA. Can be generalized, random DNA, or specialized, specific genes near phage insertion site, accidental gene delivery by virus. 3. Conjugation. Direct cell-to-cell contact. Often involves a sex pilus, a protein bridge connecting cells. Super efficient for transferring large DNA chunks, especially plasmids. The main highway for spreading antibiotic resistance. Can even occur between different species. Why this matters? This is how antibiotic resistance spreads so rapid. Bacteria can acquire resistance genes to drugs they've never even encountered, just by picking them up from neighbors. This constant genetic exchange makes antimicrobial resistance an ongoing, escalating challenge. Before we discuss how pathogens attack, let's understand what they're up against. Physical and chemical barriers. First line of defense. Intact skin. Mucous membranes lining airways and gut. Stomach acid. Low pH. Bile salts in intestines. Enzymes like lysozyme in saliva and tears. Innate cellular response. Second line. Phagocytes. White blood cells that engulf and destroy invaders. Neutrophils. PMNs. Macrophages. Mechanism. Phagolysosome fusion. Engulf the microbe and fuse it with lysosomes containing digestive enzymes. Adaptive immune system. Third line, the specialists with two arms. Humoral immunity, B cells and antibodies. B cells produce specific antibodies, IgM, IgG, IgA. Antibodies bind to pathogens, neutralizing them or marking them for destruction. Cell-mediated immunity, T cells. Cytotoxic T cells recognize and kill infected host cells. Stops pathogen replication inside cells. Prevents spread to new cells. Together, these systems form a powerful, coordinated defense. Pathogenicity, the basic ability of an organism to cause disease, yes or no. Virulence, the degree of pathogenicity, how much harm it causes, how easily. Measured by ID50, infectious dose for 50% infection. Or LD50, lethal dose for 50% death. Lower numbers equals higher virulence. Classic example, Shigella needs fewer than 100 bacteria to cause severe dysentery, while Salmonella typically needs around 100,000 organisms. That's a 1,000-fold difference in virulence, all due to different virulence factors. The stages of bacterial attack. Stage 1, entry and evasion. Capsules, the slippery shield, usually made of polysaccharides, makes bacteria slippery, harder for phagocytes to engulf. Helps hide from immune system. Example, Streptococcus pneumoniae, encapsulated strains cause disease, unencapsulated strains are harmless. Stage 2, adherence. Why it's critical, without attachment, bacteria get washed away by mucus, urine, or peristalsis. Pili/fimbriae, the grappling hooks. Protein structures on bacterial surface. Bind specifically to receptors on host cells. Example, Neisseria gonorrhoeae must have pili to cause infection, no pili, no disease. Stage 3, invasiveness. Spreading deeper into tissues using enzymes. Collagenase, breaks down collagen. Hyaluronidase, breaks down hyaluronic acid, cellular cement. These act as biological drills, dissolving barriers. Stage 4, iron sequestering. The iron war. Iron is essential for bacterial growth, but our bodies lock it up in proteins like transferrin and lactoferrin. Bacterial strategies. Siderophores, molecules with extremely high affinity for iron, molecular thieves that snatch iron from our proteins. Direct binding. Some pathogens, Neisseria, have surface receptors that directly bind our iron-carrying proteins and pull off the iron. Stage 5, antiphagocytic factors. Fighting back against immune cells. Capsules, make physical engulfment difficult. Surface proteins, interfere with phagocyte signaling. Example, Protein A on Staphylococcus aureus binds antibodies backwards, preventing effective immune tagging. Toxins, the weapons of mass destruction. Exotoxins. Characteristics. Proteins actively secreted by living bacteria. Incredibly potent, tetanus toxin, 1 microgram can be lethal. Made by both gram-positive and gram-negative bacteria. Often have specific cellular targets. AB structure. B, binding, component, attaches to host cell. A, active, component, enters cell and causes toxic effect. Examples, diphtheria toxin, stops protein synthesis. Tetanus toxin, blocks neurotransmitter release, causes paralysis. Critical advantage, because they're proteins, we can make vaccines. Treat with formaldehyde to create toxoids, inactivated but still immunogenic. Body makes antibodies against toxoid. These antibodies protect against real toxin. Basis for tetanus and diphtheria vaccines. Endotoxin, LPS. Completely different. Not secreted. It's part of the bacterial structure. Lipopolysaccharide in outer membrane of gram-negative bacteria only. Released mainly when bacteria die and break apart. The toxic component, lipid A, triggers massive systemic inflammatory response. Causes fever, hypotension, clotting problems. Leads to septic shock symptoms. The clinical paradox. treating severe gram-negative infections with antibiotics that kill bacteria quickly causes massive LPS release, potentially worsening septic shock initially before improvement. You have to kill the bacteria, but killing them releases this inflammatory trigger. Unlike exotoxins, endotoxin is heat-stable, and we can't make simple toxoid vaccines against it. Pathogenicity islands, PAIs. The attack toolkit. Distinct regions on chromosome or plasmids. Contain clusters of virulence genes. All tools needed for a particular disease mechanism. Often mobile or flanked by mobile elements. Significance. PAIs can be transferred between bacteria via horizontal gene transfer. A previously harmless bacterium could acquire a PAI and suddenly become a significant pathogen. It's like downloading a complete disease-causing software package. Viral pathogenesis. For main outcomes of viral infection. 1. Cell death, lysis. Virus replicates intensely, exhausting resources. Cell dies, often bursting to release new viruses. Example: poliovirus shuts down host DNA, RNA, and protein synthesis. 2. Transformation. Viral genetic material integrates into host DNA, disrupts normal growth controls, can lead to malignant transformation, cancer, example, HPV and cervical cancer. 3. Cell fusion, syncytia formation. Viral proteins cause infected cells to fuse with neighbors. Forms giant, abnormal multinucleated masses. Examples: herpes viruses, HIV. 4. Cytopathic effect, CPE. General visible changes in infected cells. Cell rounding, detachment, inclusion bodies. Used in labs to detect viral presence. Viral spread through the body. Initial replication, at entry site, respiratory tract, gut. Systemic spread, through lymphatic system first. Then into bloodstream, viremia, reaches distant target organs. Tropism. Viruses have preferences for specific cell types because they need particular receptors to enter cells. A virus might enter through the gut but travel via blood to infect liver cells, hepatitis, or nerve cells, polio. Vertical transmission. Mother-to-child transmission can occur 3 ways. In utero, virus crosses placenta to fetus. During delivery, baby passes through infected birth canal. After birth, through breast milk. Examples: HIV, rubella, CMV. Immune response to viruses. Cell-mediated response. Cytotoxic T lymphocytes, CTLs, and natural killer, NK, cells. Recognize and kill infected host cells. Induce apoptosis before virus factory can release new particles. Eliminate the virus production facility. Humoral response. Antibodies bind to free virus particles. Neutralize virus, preventing attachment to new cells. Mop up virus that escapes from lysed cells. Working together, these usually clear acute viral infections within weeks. And now we have important question. What is a biofilm? A biofilm isn't just a random clump of bacteria. It's a structured community. Microorganisms stick to a surface, tissue or medical device, embed themselves in a self-produced protective matrix. Matrix contains polysaccharides, proteins, and extracellular DNA. Think of it as a fortress with multiple layers of defense. Advantages of biofilm life. 1. Physical barrier. Matrix blocks disinfectants, antibiotics, and host immune components. 2. Immune evasion. Phagocytes struggle to engulf bacteria in the matrix. Antibodies can't reach targets effectively. 3. Persister cells. Slowly growing or dormant bacteria within biofilm. Many antibiotics target active growth processes. Persisters naturally resist antibiotics. Can survive treatment and regrow biofilm later. Lead to chronic or recurrent infections. Clinical impact. Massive. Approximately 65% or more of all hospital-associated infections involve biofilms. Common sites: catheters, prosthetic joints, artificial heart valves, contact lenses, chronic wounds, CF lung infections. Understanding biofilm formation and how to disrupt them is a major research focus because they're so difficult to treat. Modern diagnostics and the foundation. Advanced technologies. Modern labs have powerful new tools. NOTs, nucleic acid amplification tests like PCR detect resistance genes in hours. Whole genome sequencing, increasingly fast and affordable. MALDI-TOF MS, rapid ID based on protein profiles. But the fundamentals still matter. Here's the crucial synthesis. These advanced technologies don't replace foundational knowledge, they build on it. Why? To do MALDI-TOF, you first need a pure colony, which requires selecting the right culture media and atmosphere based on metabolic needs. To do NAAT effectively, you need to extract DNA properly. Extraction methods differ based on cell wall structure, Thick gram-positive or waxy acid-fast bacteria require harsher methods than gram-negatives. If you don't know the likely structure, extraction might fail, and your fancy molecular test gives nothing. The bottom line, new technology is powerful, but you still need foundational knowledge of microbial anatomy, chemistry, and physiology to use tools correctly and interpret results meaningfully. Key takeaways and synthesis. The ecosystem balance. Success often isn't just about bad microbes. Problems frequently arise from good microbes in the wrong place, bacteroids causing post-surgical abscess, normal competition disrupted, broad-spectrum antibiotics allowing C. diff overgrowth. Understanding your microbial allies is as important as understanding pathogens. The interconnected web. Everything we've discussed is interconnected. Identification, taxonomy, genomics, tells you which pathogen you're dealing with. Genetics, metabolism, PAIs, reveals potential for resistance and virulence. Lifestyle, biofilm formation, dictates behavior in host and treatment difficulty. Virulence factors, toxins, adherence, invasion, determine disease mechanism. Host response determines clinical presentation. This knowledge informs everything from specimen collection to diagnosis to treatment choice. The evolutionary arms race. We're in a perpetual race. Bacteria rapidly share genes through transformation, transduction, conjugation. They swap resistance plasmids and acquire PAIs. They build defensive biofilms. They adapt incredibly quickly to antibiotics. Critical questions for the future. Should we move beyond just killing bacteria to disarming them or neutralizing toxins and host responses? How do we keep our diagnostic strategies ahead of rapidly evolving resistance mechanisms? How radically must our medical research and treatment strategies change to keep pace with microbial evolution? This introduction has given you the essential foundation you need for the microbiology module. We've covered classification systems, from domains and kingdoms down to species, and why proper nomenclature matters. The major microbial groups, bacteria, viruses, fungi, protozoa, helminths, their structures and characteristics. Modern identification methods, from phenotypic characteristics to whole genome sequencing and MALDI-TOF. Normal flora, your microbial partners, where they live, and why they're beneficial. Bacterial structure, the gram stain, cell wall composition, and acid-fast bacteria. Metabolism and growth, nutritional needs, oxygen requirements, and energy production. Bacterial genetics, chromosomes, plasmids, transposons, and horizontal gene transfer. Host defenses, physical barriers, innate immunity, and adaptive immunity. Pathogenesis, how microbes cause disease through virulence factors, toxins, and PAIs. Viral mechanisms, how viruses hijack cells and spread through the body. Biofilms, the fortress strategy that makes infections so difficult to treat. The integration of old and new, why fundamental knowledge remains essential even with advanced technology. Remember, this is your foundation. In the upcoming videos, we'll dive deeper into specific bacterial families, viral groups, fungal infections, parasitology, and clinical case scenarios. But everything will build on what we've covered today. If you found this helpful, please like, subscribe, and hit that notification bell so you don't miss any videos in this series. Drop any questions in the comments below. I read them all and will address common questions in future videos. And don't forget, ASCP short notes in hematology and chemistry are now available at Amazon. Check the link in the description and wait for ASCP short notes in microbiology. This is Mohammed Hamama from ASCP Preparation Camp. Keep studying, stay focused, and I'll see you in the next video where we'll start with bacterial classification and the gram-positive cocci. Good luck with your preparation.